Welcome to all students. Myself, Om Prakash Odara, Assistant Professor in Seed Technology, Department, Faculty of Agriculture and Veterinary Science, Jyoti Vidyapeeth, Human University, Jaipur. Today, we discuss about our topic classes of seed or seed quality. Firstly, classes of seed, therefore, generally we recognize different classes of seed. Firstly, breeder seed, foundation seed, registered seed, and certified seeds. The Association of Official Seed Certifying Agency has defined these seed classes as follows. Breeder seed, the seed or vegetatively propagated materially and direct control by the originating or the spawn sponsoring breeders or institutions, which is the basic seed for recurring increase of foundation seed. Foundation seed, it is a progeny of breeder seed. The seed is stock handled to maintain specific identity and genetic purity, which may be designated or distributed and produced under careful supervisions of an agricultural experiment stations. This seed is the source of all other certified seed classes, either directly or through registered seeds. Registered seed, the progeny of the foundation seed, is so handled as to maintain its genetic identity and purity and approval and certified by certified agency. It should be of quality suitable to produce certified seeds. It is the progeny of the foundation seed. Its production is so handled to maintain genetic identity and physical purity according to standard specific for the crop being certified. It should have the minimum genetic purity of 99%. Certified seeds may be the progeny of certified seeds, provided this reproduction does not exceed two generations beyond foundation seeds, and provided that its certifications agency determines the genetic and physical purity. If not be significantly altered in case of highly self-pollinate crops, certifications of one further generations may be permitted. Certified seed produced from certified seed shall be eligible for further seed increase under certifications, except in case of highly self-pollinated crops where certifications of one further generations may be permitted. Certification takes issue once for certified seeds not eligible for further seed increase under certifications. As like paddy and wheat, certified seed produced from certified seed is eligible for certifications by nation seed corporations up to two generations from foundation seed. Foundation seed certified first and certified seconds for barley, garden bee, groundnut, soybean. Certified seed produced from certified seed is eligible for certification up to three generations for from the foundation seed. Certified first, second, and third. Certifications of certified seed produced from certified seed is not permitted for crop other than those listed. Difference between certified seeds and truthful label seeds. Certified seed certifications is voluntary, but in truthful label seeds, truthful labeling is compulsory for a notified kind of varieties. Applicable to notified kinds only, applicable to both notified and release varieties. It should satisfy both minimum field and seed standards, but in truthful level seeds tested for physical purity and generations. Seed certifications offer seed inspectors can take samples for inspections. Seed inspectors alone can take samples for checking the seed quality. Generation systems of seed multiplications. Generation systems of seed multiplications is nothing but the productions of a particular class of seed from a specific class of seed up to certified seed stage. The choice of a proper seed multiplication model is the key of further success of seed programs. This basically depends upon the rate of genetic deterioration, seed multiplication ratio, and total seed demands. Based on these factors, different seed multiplications models may be derived from each crops, and the seed multiplication agency should decide how quickly the farmers can be supplied with the seed of newly released variety. After the nucleus seed stocks has been handed over the concerned agency so that it may replace the old varieties. In view of the basic factor, the chain of seed multiplication models could be three generations model, breeder seed, after that foundation seed, and after that certified seeds. Four generation model, breeder seed, foundation seed, first foundation seed, second, and after that certified seeds. Five generations model, breeder seeds, foundation seed first, foundation seed second, certified seed first, and certified seed second. For most of the often cross-pollinated and cross-pollinated crops, 3 and 4 generations model seed usually suggested for seed multiplications. It's like castor, red gram, jute, green grams, rape, mustard, sesame, sunflower, and most of the vegetable crops. Generation systems of seed multiplications and quality controls notified varieties and hybrids. Agency, mainly agency class of seed quality control systems. Concerned breeder or sponsoring institutions और breeder himself एजेंसी होती है और इनकी जो सीट क्लासेस होती है वो न्यूक्लियर सीट होता है नो स्पेसिफाइड टैग होता है quality control systems maintenance breedings but in breeder seeds concerned breeder or sponsoring institutions or seed technologist होते हैं breed seed के लिए और इनका जो टैग का कलर होता है वो गोल्डन येलो टैग होता है इनका जो क्वालिटी कंट्रोल्स है वो ब्रीडर मॉनिटरिंग टीम्स फील्ड इंस्पेक्शन से to check genetic purity grow out test. फाउंडेशन सीड स्टेज फर्स्ट एंड सेकंड इनका जो टेक फर्स्ट होता है और उसका जो एजेंसी होता है स्टेट डिपार्टमेंट एग्रीकल्चर नेशनल सीड कॉर्पोरेशन से कोऑपरेटिव एजेंसी सेंट्रल एंड स्टेट सीड कॉर्पोरेशन प्राइवेट सेक्टर्स इसका मेनली जो स्टेट सीड सर्टिफिकेशन एजेंसी है वो फील्ड इंस्पेक्शन एंड टेस्टिंग टू चेक मिनिमम रिक्वायर्ड फिजिकल प्योरिटी एंड अदर क्वालिटी टेस्ट वो मेनली कंप्लीट करती है सो सर्टिफाइड सीड्स है उनका जो टेक कलर होता है वो जो ब्लू टेक्स होता है और ये मेनली फार्मर्स जो है उसका जो प्रोटेक्शन करता है वो ये ट्रूथफुल लेबल सीड्स होते हैं और ओपल ग्रीन का ओपल ट्रूथफुल लेबल सीड्स है उसका जो कलर है वो ओपल ग्रीन होता है nucleus and breeder seed productions the initials and full of seed obtained from the selected individuals plants of a particular variety for the purpose purifying and maintaining that varieties by the originating plant breeders and its further multiplications under his own supervisions or the supervisions of a qualified plant breeders. So, who provides breeder seed constituents the basis for all further seed productions. Mainly जो different से methods है to maintain nucleus and breeder seed in self pollinating crops तो newly released variety का maintenance किस तरीके से करते हैं और जो established varieties है उनका जो maintenance है वो किस प्रकार किया जाता है तो इस प्रकार mainly जो आपकी सीड क्लासेज ऑफ सीड है वो डिफरेंट पाई जाती है तो उसके अकॉर्डिंग mainly जो ब्रीडर सीड है या जो डिफरेंट जो एजेंसी है वो mainly इसको कंट्रोल करने का काम करती है Seed quality mainly is a basic input in agriculture. Strictly speaking, seed is an embryo, a living organism embedded in the supporting or the food storage tissue. In seed, the importance is given to the biological existence, whereas in grain, the importance is given to the supporting tissue, the economic produce. Seed is mainly defined as a ripened oil or a fertilized mature oil containing embryo which have developed after fertilizations. The dry dispersal unit or matured oils develop after fertilizations, any part or organs of plants which has the capability to regenerate into a new plants. A propagules responsible for maintaining the genetic qualities for the variety of hybrids, also known as embryo, a living organism embedded in the supporting the food storage tissue or a protective course any propagative materials miniatures plants, dormant plants, link between two generations, carrier of service material, generative part of a plant they develop into new plants. तो so मैंने definition जो है सीड के बारे में बताया गया है कि जो सीड है वो क्या होता है. However, the widely accepted definitions for a seed is a matured oil that consisting of an embryo plant together with a stock of food and all surrounding by a protective coats. तो mainly जो ये definitions है उसको maximum follow किया गया है. As per Seed Act, seed includes seed of food crops, including edibles, oil seeds, and seed of fruits and vegetables, cotton seeds, seed of cattle fodder, jute seed, seedling, tubers, bulb, rhizomes, roots, cuttings, all types of graft and other vegetative propagated materials for food crops, cattle fodders. Difference between seed and grain, it should be a viable one, but grain need not to be a viable one. So, seed, it should have maximum genetic physical purity, but grain not so as this. Seed should satisfy minimum seed certification standard, but grain no such requirements. It should be completely treated with pesticide, fungicides to protect seed against storage pest and fungicides, but in grain it should never be treated with any chemicals since used for consumption. In seed, respiration rate and other physiological and biological process should be kept at a low level during storage, but no such specific present in grain. Should be compulsory certified CO2 tools level sheets, but in grain, no such conditions in grain productions. In seed, should never be converted into grains unless warranted, but in grains can be converted as seed provided the situations warranted. In seed, it should satisfy all quality norms, but in grains, not considered all quality norms. Importance of seed is the virtual influence in crop productions because through seed only the investment made or another inputs is also less compared to other inputs, but the greater income farmer get depend upon the quality of a small quantity of seed he use. In mainly about seed is a basic for the following event in agriculture's career of new technologies. In India, for instance, the cultivations of high-yielding varieties have helped to increase food productions from 52 million tons to more than 200 million tons over a period of 50 years. Basic tool secure for food supplies, the successful implementations of the high yield varieties programs in India has led to remarkable increase in productions and to a new assessments of future development potentials. As a result, food imports from other countries have been substantially brought down in spite of the rapid population increase. The principles means to secure crop yield in less favorable production areas. The supply of good quality seed of improved varieties suitable to these areas is one of the few important immediate contributions to secure high crop yields. A medium for rapid rehabilitations of agriculture in cases of natural disaster and widespread floods and droughts in various parts of the country and elsewhere have focused attentions on these recurrent crises and the accompanying threat of famines and starvations. The establishments of national seed reserve stocks should receive high priority for meeting such natural calamities. This would provide improved seed in emergency period to productions area for rapid productions of food grains. This would supply seed to disaster regions for resolving as no seed would normally be available in such regions. Seed quality is the possessions of seed with required genetic and physical purity that is accompanied with physiological soundness and health status. Physical purity it is the cleanness of seed from other seeds, debris, inert materials, disease seed, and insect damage seeds. A seed with physical quality should have uniform size, weight, and colors, and should be free from stones, debris, and dust. Leaves, twigs, stems, flower, fruits, well without other crop seeds and inert materials. It also should be devoid say, of cereals, disease, mottles, yeah, molded, discolors, damaged, and empty seeds. The seed should be easily identifiable as a species of a specific category of a specific species. Lack of this quality characters will indirectly influence the field establishment and planting value of the seeds. These quality characters could be obtained with seed loads by proper cleaning and grading of seed processing after collections and before sowing storage. Genetic purity, it is up to type nature of the seeds. The seedling a plant a tree from the seed should resembles its mother in all aspects. This quality characters is important achieving the desired goal of raising the crop, either yield or for resistance or for desired quality factors. Physiological quality, it is the actual expressions of seed in further generations via multiplications. Physiological quality characters of seed comprise of seed generations and seed vigors. The liveliness of a seed is known as the extent. Stamina of the seed in producing elite seedling. It is the sum total of the all seed attributes that enables its regeneration of under or given conditions. Seed vigor determines the level of performance of seed or seed lot during germination and seedling resilience. Seed which performs well at sowing are termed as quality seeds and based on the degree of performance in productions. Of elite seedling, it is classified as high, mediums, and low vigor seeds. The difference in seed vigor is the differential manifestation of the deteriorative process occurring in the seeds before the ultimate loss of ability germinates. Difference in seed vigor will be expressed in rate of emergency, uniformity of emergence, and loss of seed generations. Hence, it is understood that all viable seeds need to be germinate, but all germinable seeds will be viable. Similarly, all vigorous seeds will be germinable, but all germinable seeds need not be vigorous. Physiological quality of seeds could be achieved through proper selections of seed, maturated seeds used for sowing, and by caring for quality characters during extraction, drying, and storage. Seed with good vigor is preferable for raising a good plantations. The fruits, the economic come out or the we realize after several years. Hence, selections of seed based on seed vigors is important raising perfect finalizes plantations. तो मेनली जो सीड है उसके जो डेफिनेशन है या जो क्लासेस है वो मेनली डिफरेंट मैंने क्लास आपको बताई थी और जो सीड है उसका डिफरेंट क्लास होती है उनका जो प्रोडक्शन है वो मेनली डिफरेंट डिफरेंट एजेंसीज है वो करती है प्लस जो सीड है उसकी जो क्वालिटी है वो मेनली उसकी जेनेटिक प्योरिटी है उसकी फिजिकल प्योरिटी है उसकी किलनेस है वो मेनली सीड प्रोडक्शन पे बहुत ही इम्पोर्टेंस रोल रखती है तो इस प्रकार जो मेनली सीड है उनका जो टाइप्स है जो क्लासिफिकेशन है वो अकॉर्डिंग टू एजेंसी डिफरेंट डिफरेंट होता है